Welcome to our video lecture on topic D3.2, inheritance. Today we are going to take a close look at Punnett grids, also known as Punnett squares. Our guiding questions: what patterns of inheritance exist in plants and animals, and what is the molecular basis of inheritance patterns? Lots of objectives for today. We're going to review the vocabulary of inheritance, talk about the contributions of Mendel to our understanding. We're going to use those Punnett grids, also known as Punnett squares, to predict some genotypes and some phenotypes. Those will be included in the vocabulary that we're going to look at. Some of those different inheritance patterns that we are going to complete some Punnett grids for: complete dominance and codominance and incomplete dominance. We're also going to look at some sex-linked traits. We're going to define SNPs, single nucleotide polymorphisms. We're going to wrap up with a look at phenotypic plasticity and phenology. Some of the vocabulary that we're going to start with, let's review diploid and haploid. Remember that our diploid number of chromosomes, also known as 2n number of chromosomes, this is the number that we have in our somatic or body cells. This is the normal number. This is the number of chromosomes in liver cells and brain cells and white blood cells and muscle nuclei. This is the number of chromosomes when we have complete pairs of chromosomes. So our chromosomes come in homologous pairs. We have one of those pairs from a dad and one of those from a mom. And when I have two chromosomes, one from dad and one from mom, that complete homologous pair, I have a diploid number of chromosomes. In us humans, that number is 46. Other species have other numbers, but for us humans, the diploid number is 46. During meiosis, what happens is we separate those pairs and we end up with eggs and sperm cells that only have one of each of those pairs, either a chromosome from mom or a chromosome from dad. And we're going to call that the haploid or N number of chromosomes. In us humans, again, different species have different numbers, but in us humans, that number is 23. The magic here, we have 23 chromosomes in an egg and 23 chromosomes in a sperm. These cells are known as gametes. Gametes are egg cells, and sperm cells, they are the sex cells, the cells of reproduction. They have a haploid number of cells. When fertilization occurs, these guys combine to form a zygote. So a zygote is a fertilized egg. It's an egg plus a sperm. What's pretty cool, when we have those 23 chromosomes in an egg and the 23 chromosomes in a sperm, again, when we're talking about humans, we go back to that beautiful diploid human number of 46 chromosomes. That's our zygote, our first diploid cell, which will go through mitosis to make lots more of diploid cells that eventually can grow into some full humans. Good? Good. Gregor Mendel is known as the father of genetics. He actually wanted to go to college and become a scientist, but his family couldn't afford it. And so he became a monk instead, and he worked in the gardens of the monastery and did amazing science there. What he did was pollinate pea plants. So plants have these structures called anthers. The anthers produce pollen. What's in the pollen? Sperm cells. He would collect some pollen grains and transfer it to another flower part called the stigma. When those pollen grains hit the stigma, what happens is those sperm cells travel down into this structure. Stored here are some egg cells. And so those egg cells plus those sperm cells, fertilization would occur. We'd end up with some baby plants stored in those seeds. He would plant the seeds and then see what kind of offspring, what traits in those offspring were apparent. He could sometimes self-pollinate or self-fertilize these plants, where he took the pollen, the sperm from the plant, and put it on its own flower, and so we would self-pollinate. This would lead to some clones of that parent plant. We wouldn't have a whole lot of genetic diversity there. Or he could cross-pollinate, where he would take sperm from one plant and put it on the stigma of a different plant and have a new combination of alleles, still collect the seeds after fertilization occurs, grow those plants, and see what kind of traits are evident in those offspring. We humans have been doing this for a very long time, both with plants and with animals, as we have domesticated our crops, purebred. Sometimes we call it purebred animals and plants, and that just gives us humans, the traits that we want to have in the plants and the animals that we raise for food or for work. And this is known as artificial selection. It's something to think about. As we move into evolution later in the year, we'll talk about natural selection versus artificial selection. Which one of those is evolution? Maybe both of those is evolution. It's a pretty interesting conversation that we will have. But back to Mendel. So Mendel grew those baby plants, and then he counted how many of the baby plants had different traits, and noticed that there were some dominant traits and some recessive traits. The dominants would be more apparent, more numerous in the offspring. And so he counted how many of those plants were the purplish color, how many were white, flowered, and then he reduced those numbers down to ratios. And notice that these ratios are all super close, not exactly, but super close to 3:1. We're going to talk about that 3:1 ratio in a couple more slides. What is super amazing about all this work that Mendel did, we didn't even know that chromosomes existed. So Mendel did this work. He talked about these factors that could be inherited from parent to offspring because he did this work, published his paper in 1865. We didn't even know that chromosomes existed until 1882. The hypothesis that chromosomes were involved in inheritance wasn't proposed until 1902. Mendel did this work in 1865. We didn't even know what the word gene was. Well, we didn't invent the word gene. We didn't even invent the word gene until 1909. We didn't know about A's, T's, C's, and G's until, are you ready for this? 1966. So Mendel was talking about the inheritance factors 100 years before we even knew about DNA sequences. Kind of amazing. We're going to take a look here at lots more vocabularies. So genes are sequences of DNA that code for some protein, and that protein is going to influence a trait. Alleles are different forms of genes. We talked in class about how we have hair color genes, but we could have different versions of that hair color. Those are the alleles. So maybe you have brown or black or blonde or auburn. So these are those different alleles, as different forms of the hair color gene. Alleles can be dominant or recessive. They can be other things that we'll talk about on other slides. For now, we're just going to focus on dominant versus recessive. Dominant alleles are going to mask the expression of those recessive alleles. Whereas those recessive, we're going to see only if the dominant is not there. So our recessive alleles are expressed only without the presence of those dominant alleles. We usually show dominant alleles with a capital letter, like a capital A. Recessive alleles are denoted with a lowercase letter. We're going to pick A for now. Genotype is going to be that combination of alleles that we get for a trait. And so I wrote the word pair here because we get one allele from one parent and a second allele from the other parent. So our genotypes are always going to be pairs of alleles, one that we got from mom and one that we got from dad. Homo means same. And so a homozygous genotype, we're going to have matching alleles, maybe both dominant or both recessive. Hetero means different, and so I'm going to have different alleles in my genotype, one dominant and one recessive. What is super cool is that those alleles in those genes, whether they're homozygous genotypes or heterozygous genotypes, are going to influence a trait. We're going to influence a trait, and that trait that we can actually see or measure is the phenotype. So this is the trait, the physical manifestation of the genotype. Manifestation, I can spell. Good. All right, so let's use all of those words to do this amazing question about Punnett grids. So let's say that we are doing some work with Mendel and we want to think about a homozygous recessive pea plant that has wrinkled seeds crossed with a heterozygous pea plant that has smooth seeds. So first up, we need to pick some letters for our alleles. Since this heterozygous plant has smooth seeds, we know that that dominant allele is going to be expressed. And so I'm going to pick the letter from the dominant trait. I'm going to pick a capital S for smooth. You don't have to, but this is probably the most common way that we choose letters for our alleles. My wrinkled seeds, because wrinkled is the recessive trait, I'm not going to use a W for wrinkle. I'm going to use the lowercase s. So capital S is dominant smooth, lowercase s is wrinkled recessive. My parent plants, we have homozygous recessive wrinkled seeds. That's going to be a lowercase s. Hetero, remember, means different, so the other parent is going to be capital S, lowercase s. During meiosis, what happens is those alleles get separated into different eggs and sperm. So let's say that this is the mom plant. This is the eggs in the ovules. And so the eggs can have lowercase s's, or they can have lowercase s's. Let's say that this plant is the dad plant, the sperm in the pollen grains. Those pollen grains, when those alleles get separated during meiosis, we could have a big S in a sperm cell, or we could have a lowercase s in that sperm cell. Our Punnett grid is going to show us what can happen during fertilization. We could have this big S sperm and this lowercase s egg fertilize to become a big S little s baby plant, or we could also have big S little s, or we could have a baby plant where we have a lowercase s sperm and a lowercase egg go SS. We could have, again, a zygote, a baby plant with lowercase from the sperm, lowercase from the egg. And so this is our Punnett grid. These are the potential genotypes of the offspring. We can write a ratio, and we could say that we have 2 heterozygotes to 2 homozygous recessive offsprings. We are better off reducing that down as 1 to 1. We could also talk about a ratio of the phenotypes. This is a ratio of the genotypes. The ratio of the phenotypes, we're going to have big S, little s, heterozygote. We're going to have that dominant allele mask the expression of the recessive. So we'll have 1 smooth dominant seeds to 1 wrinkled recessive seeds. We could also say that there's a 50% chance of any of those seeds being smooth, 50% chance of any of those seeds being wrinkled. Feeling good? Feeling good. Let's do another practice problem with Punnett grids. Here we're going to look at a disorder called phenylketonuria, or PKU, phenylketonuria. This is a disorder that we actually test newborn babies for, at least in developed nations. So a little heel prick pretty soon after birth, that blood gets sent off to a lab and we test to see if this baby has PKU or not. People who are homozygous recessive for phenylketonuria have a broken PA protein. So this stands for phenylalanine hydroxylase. What this protein does is it converts an amino acid that we find in protein called phenylalanine. It is converted into a different amino acid called tyrosine. If that phenylalanine hydroxylase protein is broken, then we do not convert that phenylalanine into tyrosine. That phenylalanine collects in the brain and leads to some cognitive delays. This broken phenylalanine hydroxylase protein is due to one tiny little mutation where we go from G to C here in the DNA sequence for that protein. That is going to, again, lead to the inability to convert phenylalanine into tyrosine. We can see it here in this DNA sequence where that C is kind of popped up. But luckily, if we catch it early, which is why we test those newborn babies, then we just make sure that these babies don't eat too much protein and that we don't have too much phenylalanine popping up in the system of the baby. We don't have that phenylalanine collecting in the brains, and then there are no cognitive delays. Let's talk about the inheritance pattern of this trait. It does have what we call complete dominance. Luckily, the normal version of this protein has complete dominance over the recessive, which means that in order to have the disorder, you need to be homozygous recessive. In this first example, we're looking at a parent who is homozygous dominant and another parent who is heterozygous. So since we're looking at this phenylalanine hydroxylase protein, I'm going to choose big P for my dominant normal trait and lowercase p for my recessive trait. This parent is heterozygous, so it's going to be big P, little p. This parent is homozygous dominant, big P, big P. So let's say that this is the dad. He's going to make some sperm. Those sperm are going to have one of those alleles, not both of those alleles. We could have some sperm with a big P. We could have some sperm with a little p. Our egg cells, let's say that this is the mom making some eggs. Eggs can be either big P or, oh, look at that, big P. All the eggs are going to have the normal dominant trait. So, or not trait, the normal dominant allele. When we have a cross, when egg meets sperm, we could have big P in a zygote cell. We could have big P, little p in a zygote cell. We could have another big P, or we could have big P, little p. That means that there is a 50% chance of a zygote that is homozygous dominant. There is a 50% chance of a zygote that is heterozygous. What is kind of lovely about this disorder being homozygous recessive, there is a probability of a child with PKU. There is a 0% chance of a child having PKU because this normal and this normal is going to mask that recessive. None of the offspring, none of the potential offspring from this couple are going to have PKU. So no worries about the disorder. But what if I have two carriers? Carrier is another way that we can describe heterozygous genotypes. Because if I have that allele, but I don't show it because I am heterozygous and I have that dominant allele also, I'm hiding or masking that recessive trait. I'm carrying it, but I'm not expressing it. So heterozygotes can also be called carriers. Both of the parents are carriers, both of the parents are heterozygous, and so our two genotypes are going to be big P, little p. When we do this cross, let's say that these are our eggs and these are our sperms. We can have big P, we can have big P, little p, we can have big P, little p, we can have little p, little p. Our genotypic ratios, we have one homozygous dominant. We have two heterozygotes. We have one homozygous recessive. When it comes to the phenotypes, that physical trait, all of these guys, the homozygous dominant and the heterozygotes, are going to have no PKU. They're going to be normal. This 1 out of 4 chance is going to have that disorder PKU. So the probability of a child with PKU being born to two carriers is 1 out of 1 plus 2 plus 1, total of 4. There's a 1 in 4 chance of a child having PKU. We could also write that as a 25% because we know that 1 out of 4 is 25%. There's a 25% chance of a child being born with PKU. Please make sure that we know that this Punnett square that has these 4 squares, this does not mean that this couple has exactly 4 children and exactly one of those children has PKU. This is just saying that for any child born to this couple, there's a 1 in 4 chance of PKU. There's a 25% chance of PKU. Good? Good. Because But wait, there's more. So the last two examples that we looked at were examples of complete dominance, where one allele completely masked the expression of the other one. Not all pairs of alleles work like that. We have some alleles that have what we call codominance, where literally both of them are dominant. Both of the alleles will be expressed. An example of a trait that has both complete and codominance is human blood type. So our blood types, A's, B's, AB's, and O's, comes from these super cool glycoproteins that are expressed on the membranes of our red blood cells, also known as erythrocytes. So these glycoproteins, we have a couple different versions. We can have this glycoprotein, or we can have this one that has some extra cool stuff or different extra cool stuff. What's pretty cool about the A glycoprotein and the B glycoprotein is that these two guys are what we call codominant. And so what happens is if you have both the A and B alleles, then you put both of those glycoproteins on your red blood cell. But also A and B are dominant over the O. And so if you have B or A, you don't put those O glycoproteins on your red blood cells. But if you have A and B, then you put both B's and A glycoproteins on your red blood cell, but not the O glycoproteins. Good? It's so cool and a little bit confusing. So A is dominant to O, and B is dominant to O, but A and B are codominant. Wait, we have three different alleles for this trait. We can be A or B or O. This is a little bit confusing also. So sometimes we have more than two alleles. We can have multiple alleles available to the population, but each of us is only going to inherit two of them. So even though there's three to choose from, we're only going to get two in our genotypes. Because of this weird codominance thing, we have some special notation that we need to memorize for blood type. And so instead of using the A's and the B's and the O's that I've been referencing here, we are instead going to use I's. And you're like, wait, where does I come from? We're going to use some I's. So we're going to use capital I for that dominant allele A. We're going to use another capital I for that dominant allele B. And then we're going to use, oh my gosh, words are hard. We're going to use lowercase I for that recessive allele that codes for type O. Because I have two different kinds of dominant alleles, we're going to throw some superscripts on. So capital I A is the allele that codes for type A blood. Capital IB is the allele that codes for type B blood. Lowercase I is going to code for our recessive allele that codes for O. So let's say that I've got a man with type AB blood. That means that his genotype is IAIB because he has codominant alleles A's and B's. The woman has type O blood. To get type O, you can only have the recessive alleles, so her genotype must be little I. Because if she was, let's say, IA little I, she would have type A blood because A is dominant to O. So to have type O blood, you must be homozygous recessive. This man with type AB blood can make some sperm cells that have the IA allele. He can also make sperm cells. He can't. He does. He does make sperm cells with the IB allele. The egg cells that mom is going to make, they can be little I or little I. Those are the only options for the egg cells. So when this cross occurs, one potential fertilization could be IA sperm with little I egg. We could again have that IA little I combination, or there's the potential for an IB little I or another IB little I combination. So when we get to our ratios, there is a 2-to-2, or better to reduce that down to 1-to-1 chance of genotype IA little I to IB little I. In terms of our phenotypes, the phenotypes, those physical manifestations of these genotypes, also known in this example as our blood type, we could have a 50% chance of a child with type A blood. We have a 50% chance of a child with type B blood. Good? Good. Because you guessed it. But wait, there's more again. So we talked about complete dominance, and we talked about co-dominance, and now we're talking about incomplete dominance. In complete dominance, one allele completely masked the other. Dominant masks recessive. In co-dominant, they're both equally dominant, and so you get both of those proteins being expressed. Incomplete dominance is similar to co-dominance, except instead of having a red blood cell that has A's and B's, now you get something kind of halfway between them. You get a blending of the traits. This is shown in 4 o'clock flowers, these beautiful 4 o'clock flowers, in which we have reds and whites, but the combination of red and white isn't red is dominant or white is dominant. Instead, you get a mashup of red and white, which is pink. And so if I have a red flower and a white flower and I cross them, the C red, color red, and the color white, all of the offspring, if I do that Punnett square, CR, CW, all of the offspring are CRCW. All of the offspring are CRCW, and that's pink. So a blend of red plus white makes pink. Not there's some red, and then there's some white, and there's some red, and then there's some white, but the mixture of the two gives us pink. This is known as our parent generation. This first generation of offspring is known as our first filial, the filial generation, or the F1 generation. If I take, and we do this with plants and animals, we don't do this so much with humans. If I take those babies and I have them make babies with each other, this next generation is going to be called the F2, or the second filial generation. When we put those guys together, these pink flowers are CRCW genotypes. When we do this Punnett square, we get 1/4 of the flowers are red, 1/4 are white, and 2/4 or 1/2 are pink. So pretty cool. Let's do this example. Let's say that I've got pink flowered plants and red flowered plants. To get that pink, we need to be heterozygous, that's CRCW. To be red, we have to be homozygous red, CRCR. When we do our cross, let's say that this is the pollen, this is the sperm. The sperm can have either the CR allele or the CW allele. Let's say that this was the ovule making the eggs. We can have eggs that are big CR, or once again, big C, big R. When we cross these guys, we're going to have CR, 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 CW, CR, 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 CW. And so our ratio of genotypes, we have 50%, or 1 out of 2, are big C, big R. Our other half, the other 1 out of 2, are big C, big R, big C, big W. Our phenotypic ratio would be 50% red, those guys, and 50% pink, those guys. Good? All right, because I'm going to throw another example at you next. Because you guessed it, but wait, there's more again. Everything that we've been talking about so far has been autosomal. autosomal chromosomes are those that are not our sex chromosomes. This picture is called a karyogram. It is a picture of chromosomes taken usually in late prophase or early metaphase. We humans take those chromosomes and we sort them into pairs. These pairs are homologous chromosomes. They are the ones that have the same genes in the same locations, but not necessarily the same alleles. So maybe that's hair color, that's also hair color, maybe that's blonde, and maybe that's black. We humans decided to put these chromosomes into order based on size. So chromosome pair number 1 is the biggest, and then 2, and so on and so forth until we get to pair number 22, which are these tiny little things, and then we throw on at the very end the sex chromosomes. Pair number 23 are either XX or XY for those of us who are born with a normal number of chromosomes. XX for female, XY for male, again, unless there are some interesting mutations that switch things up a little bit. How does this even work? Well, on that Y chromosome, that tiny little nugget of a Y chromosome, there is a gene called sex-determining region Y, or the SRY gene. That SRY gene codes for a protein called TDF, testes determining factor protein. If that protein is expressed because that gene is normal, then a lot of other genes are going to be altered and testicles are grown during embryonic development. If that Y chromosome is not present, that SRY gene is not present, what we end up with instead are ovaries. So the lack of TDF protein leads to the development of ovaries. There are all of these genes that are on the X chromosome and not on the Y. This gene is on the Y chromosome and not the X. So super interesting. Even though we kind of call these guys a homologous pair, clearly they are not the same. How do we end up with X's and Y's, ovaries or testicles? Well, and we're going with normal, but there are, again, lots of interesting mutations and differences, but most of the time a female will be XX and will have eggs made in ovaries. A male will be XY and will have testicles that produce sperm. Eggs, because the female is XX, the eggs will always have an X chromosome. The sperm, because the males are XY, some sperm are X and some sperm are Y. When this sperm meets this egg, we get 2 X's, and again, assuming normal development, a child with ovaries will be born. Again, XX, ovaries. Here, this X egg, this Y sperm, that child will develop testicles, and again here, testicles. And so there's a 50-50 chance of, if I could draw the zeros, of either having XX or having XY. We should have about half female children and about half male children, about half people born with ovaries, about half people born with testicles. In addition to getting testicles or ovaries, the X and Y chromosomes also lead to a bunch of other super interesting traits that we describe as being sex-linked because they are literally linked on the X and Y chromosomes. We're going to talk a lot about these X-linked traits, the traits that are on the X chromosome. We could have this unaffected father. He's got an X chromosome and a Y chromosome. We're going to call this mom a carrier. Carrier is another way for us to describe heterozygotes, especially in sex-linked traits. So mom has one dominant and one recessive allele, and so that dominant allele is going to mask that recessive trait. Dad is going to give X chromosomes to all of his daughters. So here we have some daughters. They're going to get that X chromosome from dad. They're going to get this X or that X from mom, this X or that X from mom. This daughter is heterozygous. She's a carrier like mom. This daughter is not a carrier. She's completely unaffected, homozygous dominant. This son got an X chromosome from mom. That Y chromosome from dad had nothing on it. We're going to call him, because there's nothing there. He only has half a genotype. Our prefix for half is hemizygous. And so we're going to say that his genotype is hemizygous dominant. This son got this X chromosome from mom, got that Y chromosome from dad. Only this allele for this trait is here. It is the recessive version, and so this son will be hemizygous recessive. Hemi, just half a genotype. We use X's and Y's to show that these traits are linked on the X and Y chromosomes, and so I could show X big H, X little h for these hemizygous genotypes of the males, and we do not put any letters on those Y's because there is no allele on those Y's, and so those guys just stay totally blank. Those females, though, can certainly be homozygous, and we could be homozygous dominant, we could be homozygous recessive. Heterozygotes, our carriers, are going to have one of each allele, one dominant, one recessive. I've been using the letter H because I'm already thinking about this trait, hemophilia, which is going to be the example that we're going to look at here. Hemophilia is a disorder of the platelets, the clotting factors in blood. People with hemophilia can get a tiny little cut and bleed and bleed and bleed because their blood does not clot appropriately. So let's predict the ratio of phenotypes of potential offspring of a mother who is not a carrier for hemophilia and a father who has hemophilia. If mom is not a carrier, that means that she must be homozygous dominant, and so mom's genotype, she's XX because she's female, because she's totally normal, homozygous dominant, she is X big H, X big H. Dad is XY, and he does have hemophilia, so his X has that hemophilia recessive allele. His Y has no allele. So let's see what happens when egg meets sperm. Mom's eggs, dad's sperm. So mom is going to give X chromosomes to all of her children. She's going to give normal, dominant alleles to all of her children. Dad is going to give his daughters an X little h, X little h. Dad's going to give his sons those Y chromosomes. And so what we have is 50% chance of having female children. All of the female children will be carriers. We have 50% chance of having a male child that's totally normal. So we have a 1 in 2 chance. Half of the children should be female carriers, and half of the children should be male, totally normal. So any given child has a 50-50 chance, a 1 in 2 chance of being either female carrier or normal male. And I use normal in terms of hemophilia, not in terms of all the other amazingly quirky things about being human. Let's say this other example. Now I have a mom who is a carrier for hemophilia and a father who does not have hemophilia. Carrier, again, is our other way of saying that mom is heterozygous. She carries that allele for hemophilia, but she does not herself have hemophilia. Dad, XY, does not have hemophilia, so he has that normal allele. And when we have egg meets sperm, we could have 2 X's that are both big H's. We could have 2 X's where one is a big H and one's a little h. We could have an X and a Y where that X has a big H. We could have an X and a Y where that X has a little h. So there's a 1 in 4 chance of having a female child that is homozygous dominant, totally normal. There is a 1 in 4 chance of having a female child who is a carrier. There is a 1 in 4 chance of having a male child who is normal. There is a 1 in 4 chance of having a male child who does have hemophilia. Notice here that if I'm looking at just the female children, 0% of them are hemophiliacs. If I'm looking at the male children that are possible from this couple, half of them could have hemophilia. Good, good. All of these different alleles that we've been talking about, these different forms of genes, are the consequence of mutations and SNPs. Mutations change DNA sequences and lead to the formation of SNPs. SNP, we talked about this a couple of units ago, are single nucleotide polymorphisms. SNPs, single nucleotide polymorphisms. Single, literally one, nucleotides are our A's, T's, C's, and G's. Polymorphisms, poly means many, morph is shape or form. So we're talking about many different possibilities for these nucleotides in some location in our genes. Sickle cell anemia is one that we've been talking about already. So this is our normal DNA sequence. Sometimes that T gets flipped into an A. That means that our messenger RNA transcript is going to have an A, or it's going to have a U where it should have an A. That means that in our protein, our hemoglobin protein, where we should have glutamic acid, we now have valine, and that's going to lead to some clumpy hemoglobin and then to our sickled cells, our sickled red blood cells. This is the consequence of a mutation of a single nucleotide polymorphism. This is one allele. This is a different allele. We can also have, oh my gosh, lots and lots of different alleles. These are just some chosen nucleotides for the protein that codes for cytochrome C oxidase, which is this protein complex right here in the electron transport chain of the mitochondria. It's involved in aerobic cellular respiration. Cytochrome C is this protein here. It's going to give some electrons to this guy, which is the cytochrome C oxidase. This guy is also in charge of throwing hydrogens and electrons onto oxygen to make the water. Remember that oxygen is the final electron acceptor. At the end, we're talking about the end of the electron transport chain, so that we can make lots of ATP. These are lots and lots of different bird species, and notice that they have lots in common, but there are some mutations, and so these again are examples of single nucleotide polymorphisms, lots and lots of alleles amongst these bird species for this cytochrome C oxidase protein. What's crazy cool about these phenotypes, these traits that we've been talking about, is that they can sometimes be influenced by the environment. We've been talking about some traits that are exclusively controlled by our genetics, like ABO blood type. If you are type O or type A, this is all about those alleles that you have. Color blindness also is controlled exclusively by genetics. There are some traits that we have that are exclusively environmental. If you're a cellist, it's not because you had the gene that made you a cellist. It's because you learned how to play the cello. If you have knee surgery, that trait, that scar that you have is not the result of your genetics. It is the result of your environment that led to some knee surgery. Sometimes, though, we have super cool mashups between genes and the environment. Height is controlled in part by genetics, but it also is related to nutrition and how our muscles are developed. And so height isn't just about genetics, it is also about the environment. The development of cancer is also an interaction between genes and the environment. We talked in our last video lecture about our genes that are tumor suppressor genes and our proto-oncogenes, and those genetics can lead to the development of cancer, but the environment also is going to influence those genes and turn them into cancerous genes instead of the not cancerous ones. The environment can also turn on and turn off different genes. So this idea of phenotypic plasticity, changing our phenotypes due to the environment, is pretty cool. So bacteria have this super cool thing called a lac operon. It is this cluster of genes. These genes are going to code for the lactase enzyme that breaks down lactose. If there is no lactose in the presence of the bacteria, why would it make this enzyme if there isn't even any lactose to digest? And so when there is no lactose in the bacteria's environment, this set of genes is shut down. This repressor is like, nope, don't do it. But when there is lactose present in the environment, then we take that repressor off and we do make all of those genes, and then you have the lactase to break down the lactose. So we can turn on and turn off genes. We can have the phenotype or don't have the phenotype depending on the environment. This also occurs in plants and other organisms based on seasons. And so temperatures and sunlight can influence the expression of different kinds of genes. We can activate and inactivate genes. We can have phenotypes being expressed or not being expressed due to temperatures and the amount of sunlight. And so we can turn on, let's go ahead and grow some leaves, or it's too cold. No, don't grow the leaves. And so this, again, plasticity. This particular version of plasticity based on seasons or time is known as phenology. Super, super cool, but also kind of scary when you start thinking about how climate change might be changing these temperatures and kind of messing with that phenology of the plants. And on that not super happy note, we have arrived at the end of our video lecture. We did so much vocabulary. We talked about Mendel. We used those Punnett grids, complete dominance like we have with PKU, codominance like we have with blood type. Remember to use the eyes when we're talking about the genotypes of blood type. Incomplete dominance, those super pretty pink flowers that we get when we cross reds and whites. Sex-linked inheritance like we have with the hemophilia. Remember that we use X's and Y's and then our superscripts on the X's. SNPs are single nucleotide polymorphisms, the formation of different alleles due to mutation. Phenotypic plasticity. We can change those phenotypes depending on environmental influences. Phenology is related to seasons, turning on and off traits due to the climate. Good work today.